Hello and welcome to Market Open Direct Connect, a webinar series for companies to tell their story, why it's compelling and why it's an opportunity to invest. I'm your host, Stuart Walters, and we're joined here today by Dr. Dallimore, Managing Director and CEO of Argenica Therapeutics, ASX code AGN. Dr. Dallimore will provide an update on the company's phase 2 clinical trial program in acute ischemic stroke and will participate in a Q&A post-presentation. If you do have any questions for Liz, you can submit them via the Q&A tab at the bottom of your screen. It's time to introduce our guest. Liz, welcome to Direct Connect and over to you.
Thanks so much, Stuart. And thanks everyone, I guess, for joining the webinar. And I really just wanted to, it is my first sort of direct webinar through the company to potential shareholders, current shareholders, investors, etc. So delighted to just have a little bit more time and the opportunity for people to ask questions as well, but a bit more time to delve into the phase 2 clinical trial, which is obviously in full swing and up to 20 patients recruited now, which is has exceeded our expectations in terms of timeframes for patient recruitment. So we're absolutely delighted where the trial is going. So I'll just, so you can see my screen there, the slides, yeah? All good?
Yes.
So I'll just run into just a quick reminder. I'm sure most of the people on the webinar now are hopefully familiar with the story of Argenica, but just a quick sort of refresher as to our lead drug candidate being ARG-007. It is a peptide drug. We have the luxury of having huge amounts of preclinical efficacy data, predominantly in ischemic stroke, which is our lead indication, and some really interesting data now to delve into the mechanism of action, so how that drug is working. And so the main focus of this webinar is on that phase 2 trial, but also happy to take questions on our other indications in the clinic, which are shown there. So just a quick overview of company metrics. So we are well funded at the moment, having completed our $12 million placement a couple of months ago, which is fantastic. So funded well and truly to the end of our phase 2 clinical trial. And now just teetering just over that 100 mil market cap as a company, which is fantastic as well. So I just want to quickly delve a little bit into talking a bit more about the mechanism of action of the drug and some of the data that we've generated, not only on our drug, but also competitor drugs in the clinic, to really show the point of difference of ARG-007 and how this drug is working in acute ischemic stroke. So for those of you not aware of what happens in an ischemic stroke, so these are strokes caused by a clot in a blood vessel in the brain. So they make up around 85% of all strokes. The other type of stroke are hemorrhagic strokes, which are caused by a bleed on the brain. So when you have a clot in a blood vessel in a brain, it prevents blood flow, obviously, to that part of the brain, and the blood flow carries oxygen, which is absolutely critical for brain cells to survive. And these brain cells only last around 3 seconds without this oxygen supply that's delivered by the blood. So what happens when that blood flow is restricted to that part of the brain? There is a neurotransmitter, so a chemical in the brain that is overexcreted. This neurotransmitter is called glutamate. And that glutamate overexcretion sits outside the cell in what's known as the extracellular matrix. So the bit of fluid that's around, that sits around the brain cells. This glutamate then actually triggers a whole bunch of these little channels, or little what we call receptors, on the surface of brain cells and causes them to open up, letting calcium flood into these brain cells. So this really, that calcium influx, that opening of these receptors, is really the first critical point of brain injury that happens post-stroke. This is known as exocytotoxicity, and this happens in not only stroke, but all the other indications that we're actually looking at as well. So when you get this calcium influx into brain cells, it causes brain cells to almost implode on themselves, which results in more glutamate being released, and the cycle just keeps going. And so this is why in stroke and other types of brain injury, you see this sort of cascading out of brain cell death that's happening. And we can see that on imaging. And I want to show some images later on as to why the sort of imaging is so important. But essentially what we're trying to do in terms of stopping that cascade out of brain cell death. So what we know about our drug now is that it actually stops, it's pulling those little channels or all those little ports on the brain cells into the brain cells themselves. So stopping the ability for that calcium to flow into that brain. So stopping that exocytotoxicity. And so what we've done is we've actually obviously looked at our drug and the ability to stop this calcium influx. And we do this in cell culture. So we take brain cells and we put them in little Petri dishes and we actually flood those brain cells with glutamate to reflect that condition that is happening in the brain. So that upregulation of glutamate that's happening around the brain cells. And then we see how well those cells survive. So we look at that cell viability. And so essentially what this data is showing is that when you flood these cells with glutamate, they start dying pretty quickly. So the cell viability is pretty low. But when you administer our drug, so in scientific literature, we call it R18D. So this is the Arge007 drug. When you actually put our drug in with these cells, it protects them almost up to 80 to 100%. And we can see a bit of a dose response. And we've compared this to competitor drugs that are currently in the clinic. And so what we know now is our drug is the only drug that is currently in the clinic that seems to be blocking this key process, this exocytotoxicity that is happening in stroke. So this is really exciting. The closest competitor we have at the moment is the Aptol drug, which is blocking inflammation. So really exciting to see that our drug seems to be the only one that's really working upstream at that critical point of brain cell death following ischemic stroke. So we obviously look at how this works in animal models. That's allowed us to actually get into our clinical trials. So just reiterating, we've seen some pretty compelling results in our preclinical model. In our non-human primate, we see that 70% reduction in brain injury. And a number of other critical preclinical studies that are really required to commercialize this drug. So showing that it doesn't exacerbate bleeding in hemorrhagic stroke, showing that it can be administered with current standard of care clot dissolving drugs, and showing that it's safe in healthy human volunteers. So just quickly, a refresher around what we were actually doing in our phase 2 trial. This is known as the Sean trial. This is a name that was not chosen by me, but obviously aligns very closely with Arge007. So the clinicians have a James Bond theme going on, which is absolutely fine. Although some people do say it's not really a license to kill, it's a license to survive. So that's what we're going with. So we are activating 10 hospitals around Australia to recruit 92 patients into the trial. Really that number of 92 patients has come from analysing all our preclinical data, in particular the non-human primate data, but also looking at other comparative clinical studies that have gone ahead of us. One study in particular called Escape NA and Escape NA Next. So looking at sort of what type of brain injury reduction those studies were getting. So the statisticians took all that data and essentially came up with 92 patients would be the right number to see not only an effect around safety, but hopefully start seeing these signals of efficacy in the trial. The trial is double-blinded, meaning that we don't know which patients are receiving a saline placebo and which are receiving our drug, and we won't know that until the very end of the trial when we actually lock down the data and unblind it all. We are taking the highest dose from our phase 1 clinical trial into this phase 2 trial, really to give us the best chance to see efficacy. And I'll come to a bit more about the trial design and why that is so critical and why we spent so long on the trial design and why we're really hoping that this trial design will give us this signal of efficacy. So we have now 8 Australian hospitals activated across the country. The final 2 are likely to come on board next week. So we'll have a really good sense once all the sites are activated how well that recruitment is going. But that should say 20 patients recruited. So we've got 20 patients now per the announcement this morning that have now actually been dosed in the trial, which is absolutely fantastic. And that's come from only 5 of the sites that have been activated. So the trial really is progressing, I guess, beyond our expectations in terms of the timeframes for that patient recruitment. So we are ahead of schedule. We had predicted that we'd finish dosing in Q4 next calendar year, and we are ahead of that schedule at this stage. Once we have all sites activated, I will be able to give a bit more of an overview in terms of what the timeline to complete dosing will be. So the final 2 hospitals that we are looking that will activate next week are Monash Health and Gold Coast University. Both of these have had their site initiation visits, so it's just a bit of final paperwork to get them activated. Once the sites are activated, it does take a couple of weeks for the clinical trial teams to get familiar with the trial protocol, to really understand the screening of the patients. Once they do their first patient, we find then they get sort of quite well versed in how to recruit. I've just come off a call with all the principal investigators across all the sites, and so far the feedback on the trial has been extremely positive. There's been no issues with consenting patients into the trial. They're consenting in for a standard of care procedure anyway. So there's some teething problems with data entry and the system, as there always is in these types of trials. But in terms of the ability for these sites to recruit, they're really positive. They're really positive around the trial protocol and how we're engaging with each of the sites, which is fantastic. So I just want to run through just a bit more detail on the clinical trial protocol and what we're doing and why it's kind of so critical. So in this phase 2 trial, we really are looking at safety. That this is the primary endpoint of the trial, and really the biggest hurdle that we do have to overcome from a commercialization point of view and getting a drug approved. Because in neurology, in neuroscience, when you deliver a drug to a patient that has a neurological injury or condition, the first thing you need to show is that the drug does no harm. So it's quite different from oncology, where you obviously want a drug to have a toxic effect. In neurology, you really do need to show that the drug does no harm to these patients. So this is the highest hurdle that we do need to overcome. In something like ischemic stroke, where there are no drugs on the market that can actually reduce brain injury following stroke, the efficacy hurdle then is a bit lower, because you're really just trying to see some efficacy. So we've really designed this trial around safety, but what we also want to do from a commercial point of view is to find a signal of efficacy to really give us a sense of what a phase 3 clinical trial would look like, how that needs to be designed. So because safety is the primary endpoint, what the regulatory authorities, including the FDA, require us to do is to show that the drug is safe in acute ischemic stroke patients only. So to do that, we do need to make sure that we are administering the drug only to acute ischemic stroke patients. In terms of the clinical workflow that we have in these patients, so once someone has a stroke and the ambulance arrives, or the Royal Flying Doctors, helicopter, whatever, there's very little that the paramedic can actually do for that patient. So the idea is to get them into a large tertiary stroke care unit in one of the metro cities as soon as possible. Once they're in emergency, they will have a scan diagnose the type of stroke. So this is really where the screening starts. Although, you know, talking to the PIs, they're already screening patients as they're coming in the ambulance or as the helicopter's landing on the helipad at these hospitals. So they're already starting to look at, is this patient potentially eligible for this clinical trial, which is for us is fantastic that the screening is starting so quickly. So once they're actually, then it's a confirmed diagnosis of a large vessel occlusion, so a clot in a large vessel in the brain, and that patient is eligible for thrombectomy, then we know that we can deliver our drug to that patient. In terms of those safety endpoints, we are looking at obviously mortality rates. So obviously wanting to show that our drug does not cause any mortality, and then the frequency of any adverse or serious adverse events at a number of different time points, as shown on the slide. We do have a secondary endpoint that is an efficacy secondary endpoint around infarct volume reduction. So this is just the volume of brain injury that we actually see on imaging. And this is done between the treatment group and the placebo group at around 48 hours post that stroke. So really seeing post-stroke if our drug is having an impact on that infarct volume reduction. So really what I want to show here in these brain images, and these are not images from our trial, these are from a paper that's referenced on the slide, just to give you a sense of what we are trying to see in this trial. So patients will come into the emergency department. They have that initial scan, as I said, and this is really the baseline image that we're taking. That baseline image gives the neurologist a lot of information around the severity of the stroke, the location of the stroke, the likelihood of that patient sort of having a good result post thrombectomy. So thrombectomy is that mechanical procedure that aspirates the clot out of the brain. But what we know about thrombectomy now is that it actually causes a secondary injury, this reperfusion injury. So quickly removing a clot from a blood vessel of the brain and blood rushing back in the brain can actually cause what we call penumbra, or still salvageable brain tissue, to start to die. And so you can see this on this imaging here. So where we have the yellow image, the yellow area, this is brain tissue that is still alive. We call it the penumbra. It's still salvageable brain tissue. But for some reason, post-mechanical thrombectomy, MT, it actually starts to die. And we can see that it's causing additional infarct. So we've designed the trial knowing that we have to deliver our drug in the emergency department, so as opposed to the ambulance, which would be better, giving it sort of closer to the stroke. Knowing we have to deliver it in the emergency department, what we decided to do is only include patients that are actually having this mechanical thrombectomy procedure to give the drug the best chance of working on this reperfusion injury. So protecting this still yellow area. So really trying to draw out that efficacy signal in this trial. So really what we then need to do, and one of the key exclusion criteria is that we're working really closely with the principal investigators on the trial, is what's called an aspect score. So an aspect score, if a patient has an aspect score of 0 to 5, then they are excluded from being eligible for the trial. And the reason for this is the lower the aspect score, the more brain injury that you see on the imaging. So someone with an aspect score of 10, pretty normal brain, looking pretty good. So still a lot of salvageable brain tissue for our drug to work. Then we sort of work down. But if we see an aspect score of 5, all the way down to 0, there's quite extensive brain injury. That brain cells are already dead, and there's really nothing our drug could do. So we wanted to make sure we had a population of clinical trial participants in this trial that had that still salvageable brain tissue for our drug to work on. So that's a really key aspect of our clinical trial design, to give us the best chance of seeing that efficacy signal. So in terms of the near-term catalyst and what we will be reporting on the trial, so as I said in the announcement this morning, we've recruited 20 patients. When we get to 23 patients, we will send all the data to our data safety monitoring board, who will really analyse all the data, make sure everything is looking okay, look at any of the adverse events, confirm that they're not related to drug administration, and give us the go-ahead essentially to keep going with the trial. We don't stop the trial as that DSMB data is being collated to send to the DSB, or while they're deliberating the data, the trial keeps continuing. And throughout the trial, I'll continue just to give updates around recruitment, the outcomes of the DSMB as well. And we are then looking to put in an investigational new drug application to the FDA. So we've almost completed the last of our IND enabling studies. So this is just some small in vitro studies that we had to complete. We've started compiling the information to put into that investigational new drug application, which we will aim to get to submit to the FDA towards the end of this year. Once we have that open IND, it does then allow us to actually have look at potential regulatory incentives that we can access. So things like the fast track regulatory application. So this allows us greater access to the FDA. We get fast track review of any of our clinical data, of our program of work. We'll also look at the potential for things like breakthrough therapy and the like, which again gives us that greater interaction with the FDA as we come to think about a potential phase 3 clinical trial. And we will also obviously progress the other indications in HAE, TBI and Alzheimer's disease. Which I might just quickly touch on. So, and just to sort of reiterate those key investment highlights as well. So solving those large unmet needs, huge amounts of preclinical data, which does make my life easier when we're having discussions with big pharma and the like. Exciting near-term catalysts, both in the phase 2 clinical trial as the recruitment progresses faster than expected, but also in the other indications. And then just quickly touching on partnering opportunities. So we continue to have discussions with potential partners to really look at partnering post our phase 2. The feedback to date has been positive, but really what a lot of these partners want to see is that phase 2 data. So we'll continue to progress those discussions and we'll obviously notify the market if anything material arises from those discussions. So in terms of the other indications, just touching on quickly refreshing people's memories around the opportunity in traumatic brain injury. So the data that we're getting out both from our rodent studies as well as our larger animal studies, and we use the ferret model, which is a really good model for TBI, is we're seeing some really exciting, compelling data in these 2 animal models. So we will have further data coming out in both rodent studies and ferret studies over the next 12 months. And then just quickly touching on HIE as well. So we're doing large animal studies in piglets and lambs. In HIE, we do have the benefit of orphan drug designation and rare pediatric disease designation for this indication. I think, Stu, there's a question around that as well. So I might stop there and just turn to some of the questions that came through the Investor Hub, but also if there's any other questions online.
Liz, thank you. Fantastic presentation. As mentioned, we do have a couple of questions or a number of questions that come in. Have there been any issues with consenting patients into the trial so far?
No. So as I mentioned, I had just come off a call with all of the principal investigators across all the hospitals. So they've had no issues with consent. We've only had one patient actually not consent. He was quite a young patient whose parents decided it wasn't sort of something that they wanted to do. And I guess, you know, I completely understand that it's a pretty confronting situation, especially as a young person having a serious stroke. So, but apart from that, it's all been pretty smooth sailing in terms of consent.
How are partnering discussions progressing?
Yeah, so I'd headed to bio in June, and it was quite interesting. I've been going to bio for a number of years, and it's, going from sort of a preclinical company now into having started a phase 2 clinical trial, and just seeing the increase in engagement in terms of those partnering meetings. So they're all still early stage. It does take a little bit of time to obviously get these things moving. The partners that we're in discussions with are obviously really keen to understand the outcome of the phase 2 clinical trial. But one thing I will say, it's been really refreshing to see the shift into looking at assets in neurology. There is a marked difference from, sort of four to 10 years ago, when neurology was a bit on the back burner, but it's looking at CNS assets is something that's sort of front and center of a lot of not only big pharma but mid-tier pharma. So we're in a good position, and we've got some really fantastic, compelling data to be able to share with them.
And when do you expect an outcome from the next DSMBs?
Yeah, so we obviously only need to recruit 3 more patients to be able to activate that DSMB. So we're now in the process of collecting all the data, looking at when we can actually lock in that meeting. So the DSMB is made up of independent neurologists who are sort of all over the globe. So it takes a little bit of time to coordinate their calendars to get them all together. So we're in the process of doing that. And so it would probably be around late August, I imagine, that we would sort of look to be able to actually lock them down and have that meeting and compile the data.
Is the current trial underway particularly challenging because you can only trial it after someone's had a stroke?
Yeah, look, that does bring challenges, but it also makes things easier as well. So I don't actually have to go out and promote the trial really to potential patients, or do any sort of social media campaigns or anything like that. Obviously the patients come to the trial site regardless. So there is quite an extensive number of patients in Australia, unfortunately, that are having large vessel occlusions coming into these metro hospitals. So the most recent audit, there's 2,200 people across Australia presenting every year to stroke care units that are eligible for thrombectomy. So yeah, it's no actual shortage of patients. So really the thing that we are really instilling into the clinical trial sites is getting the right type of patients that is going to give us the best chance of hopefully seeing that efficacy signal. So really taking the time to look at the brain imaging, that's still salvageable brain tissue, and getting a breadth of patients in terms of ages as well. We don't want sort of all 90-year-old patients. So we've got now a good breadth in those 20 patients, the youngest being 45. And, we've got only a couple in their 90s as well, and an even split between male and female. So that's really what we're doing is making sure that we've sort of got a good representation as well. So no, not particularly challenging. So the biggest bugbear that the PIs are having at the moment is some computer issues. So some technical issues. So in data entry, but apart from that, it seems to be going well.
Good news. Can you explain what the priority review voucher program is and why it's something Argenica might benefit from?
Yeah, so the rare pediatric disease designation comes with a priority review voucher program attached to it. So when you apply for orphan drug designation for a rare pediatric condition, you have the ability to also ask the FDA if you can get a rare pediatric disease designation. So what that means is that once you have a drug approved in that rare pediatric disease designation, you have the ability to actually get a voucher. That voucher can then be used on subsequent drug development. So whereas we're valuable, I guess, for a small company, is you can actually sell that voucher on. So sales prices of those vouchers do vary, but it can be sort of in the order of 100 to 150 million US dollars. Because if you think about selling it to a large pharma company, if they can rein in the review time from the FDA by 6 months, they've got a blockbuster drug, where they may be making $500,000, $500 million, you know, per quarter on that drug in terms of sales, then, you know, paying $100 million to get 6 months more of sales is something that's definitely worth their while. So that's where it really sort of benefits small companies. One thing I will say though about that priority review voucher is that we do need to get the HIE indication approved before stroke. So we are just at the moment working with the FDA around some nuances of that if we get the stroke drug approved first. So yeah, just wanted to flag that.
How long is a patient monitored post-dosage and the frequency of this analysis?
Yeah, so in those time frames that I put, so we are doing regular reviews of that patient. What we're trying to do also in this trial, which I sort of didn't touch on, is actually gather a lot of data around the exploratory endpoints. So we do safety time points, so obviously looking at any adverse events out to 90 days. But we're also then collecting data around imaging at those time points and also looking at some key functional assessments, in particular at 30 days and then 90 days. So when we come to do the phase 3 trial, we'll switch it around, and that 90-day functional assessment will actually be our primary endpoint in our phase 3 clinical trial. So we really wanted to collect all that data as well in these patients. So, and looking through the data with the PIs this morning, you know, they're doing such a fantastic job in collecting all that data. It is a lot. It's more data than they would typically collect, obviously, with standard of care, but they're all doing it and they're all entering it into the system. So post this trial, we'll have so much data, which will really allow us to search for the signal and design the right phase 3 trial to get us that efficacy outcome.
Do you have any idea about the ultimate price per dose of treatment that would be charged to the patient or the insurer?
Yeah, so we've actually done quite a lot of work on pricing with both US neurologists, US payers, insurers, and hospitals. So the way that the reimbursement in stroke works for a neuroprotection drug is it comes under what's known as a DRG code. So at the moment, there's a reimbursed amount of $30,000 per patient per treatment. So a neuroprotective drug would come under that, and then the idea would be that you sort of show how well that drug's doing in terms of reducing functional deficits for these patients, and then that DRG code increases. So that's sort of how the reimbursement works. So then we've done work on, well, how much would a insurer pay? So they'll pay up to that DRG code. And so we've sort of priced it at the moment at around $10,000. That seems to be the price that you know, that the insurers are sort of saying, okay. So then we went to the hospitals and said, okay, if we breach that cap of that DRG code and we actually charge more, if we charge 20,000, would the hospitals make up the difference? And it was a resounding, absolutely, we would make up the difference, and we wouldn't see a $20,000 sort of dollar price point as being particularly, you know, onerous. And because that means if they can get their patient out of hospital quicker, then that is a massive cost saving to the healthcare system in the US. So at the moment, bottom price around 10,000, but given how we the benefits of a neuroprotective drug, it's likely to be priced higher.
Is it medically obvious after day 2 or 3 if someone's had a placebo?
That is a good question. So we don't see that imaging data at the moment, but the DSMB does have access to that data, but it is all blinded. So we don't really, you know, we're not sort of scrutinizing it at this stage. Obviously, we hope that we do see a reduction, an obvious reduction in that infarct volume on the imaging. But what we will be doing, like to the naked eye, to quantify it can be more challenging. So we will be overlaying AI technology on all those imaging files to really understand how much of a reduction in that infarct we are actually seeing of the placebo versus the treatment group. But 48 hours in terms of that imaging, it's enough for the drug, you know, to work. That's what we've modelled in all our preclinical studies. So that's that. In the primate study, we see a 67% reduction at 24 hours and 24-hour imaging post-stroke.
Can you explain or advise the level of due diligence that both MLC Investments and IOOF Investments have done on Argenica, and any comments they've made regarding the Arge 007?
Look, yeah, you'd probably have to ask them. So that particular fund has done a significant amount of due diligence on the company, as I would expect, obviously, any institutional grade investor and any investor to do, really. So it's been a lot of discussions with the company, visiting the lab. So yeah, significant amount of due diligence and many questions throughout the process. But in terms of what they're doing in the background, obviously I'm not privy to any of that due diligence process.
Okay, we've had one more come in. If you pass the phase 2 trial, how quickly do you anticipate it will take before announcing a partnering agreement? And are you already in advanced discussions with multiple big pharma companies?
Yeah, look, I wouldn't say we're in advanced discussions. We are in discussions, as I said, you know, sort of post the bio partnering conference. We are following up with a number of potential partners. In terms of how long post the phase 2, at this stage, it's hard to say. We're just at the process of trying to obviously get CDAs in place, look at them to sort of do some due diligence on the data that we already have. But yeah, it may, you know, in terms of sort of partnering post phase 2, there's a whole raft of different ways that we could potentially look at it. So the main thing is to keep the program going and get into that phase 3 as quickly as possible. So, you know, we can ideally get this drug on the market as soon as possible. So paralleling that with the partnering discussions is sort of the strategy of the company at the moment.
Liz, fantastic presentation and some great questions that have come in. So thank you for answering those. For more information about Argenica Therapeutics, head to the company website, argenica.com.au. Sign up to the Argenica Investor Hub page, and you can follow the company on their social media platforms. Thank you to everyone for attending today's webinar. Another fantastic presentation by Dr. Liz Dallimore. If you would like to hear from a specific company, please send me an email, stuart@marketopen.com.au, and I'll reach out to the company on your behalf. Liz, thanks for your time today. Keep up the great work and look forward to chatting again soon.
Thanks, Stu. See ya.